Hello, BISC130. This is the beginning of recorded lecture 2.2, starting the chapter on metabolism. So, as we get started, remember, this class has no prerequisites. So, if you've ever taken a physics class in the past, all of this stuff is going to be, review for you. But to get everyone up on the same level here, let's pretend we're a physics class for a few minutes and just review some basics. We will need to get these fundamentals down to understand, stuff going on in biology. So let's start by defining energy. We could define energy as simply the ability to do work, to make stuff happen, to change things in the world. Simple definition, the ability to do work. We could divide energy into two types. Let's start with potential energy. Potential energy is a type of energy that can be thought of as stored energy. Nothing is moving, nothing is happening, but there's the potential for it to do so. Classic example from a physics textbook, kid at top of slide. So again, nothing is moving here, but you know that there's the potential to go down this slide. Another more grand example, perhaps, is a dam holding up a bunch of water. All this water here in the reservoir really wants to flow downhill, because gravity, but the dam is holding it back. There's a lot of potential in the water up here, but nothing's moving right now. So what does this look like in biology? What does this look like on a chemical level? Well, this potential energy, this stored energy, includes a type of energy called chemical energy, which is energy in chemical bonds. So, we've seen molecules like this in the past. Just by having these covalent bonds between these atoms, this is an energy storage molecule. So chemical energy is energy that is stored in these chemical bonds. And again, I've written this exact same thing in an earlier chapter, but breaking chemical bonds releases energy. So you could have chemical energy, chemical potential energy, just by having chemical bonds. So that's what things look like in chemistry and biology. So what about the other type of energy? So this is stored energy. All this stuff is potential energy. The other type of energy is called kinetic energy, which is the energy of motion. So kinetic energy would be, kid going down the slide. That's movement, that's motion. Water flowing downhill, whether it's through a dam or whether it's in a waterfall, it's going where it wants to go. That's kinetic energy. Again, these are good physics examples, but what does this look like in biology? Well, I guess this isn't really biology, but it's taking these biological molecules, burning them, and making car go fast. So yeah, this is movement. This is kinetic energy. Importantly, kinetic energy does include heat. So heat on an atomic level is moving molecules. So if it's really hot outside, the molecules in the air surrounding you are moving quickly. At a cooler temperature, the molecules are just going to be moving more slowly. So, we don't really think of heat as movement, but heat really is a type of kinetic energy. We're talking about movement of these molecules. Okay, so the other physics thing we have to do is to talk about the laws of thermodynamics, which sounds incredibly intimidating, but I'm actually crossing out the S here. We're just going to talk about one of these that I think is important, the first law of thermodynamics. And again, this sounds way more intimidating than it actually is. The first law of thermodynamics simply states energy cannot be created or destroyed. That's it. That's all the first law is. You can't make energy out of nowhere, and you can't completely destroy energy either. All we can do with energy is shuffle it around. We can convert energy from one form to another. So, yeah, we can convert potential energy to kinetic energy, and we can convert potential energy to kinetic energy. And yep, you can see where I'm going here. We can convert this chemical potential energy, burning this in an engine, into kinetic energy. And if we want to turn more to a biological side of things, we could take chemical energy in our food and convert that to, the kinetic energy of, running around, powering our muscles. And if we want to go outside of animals to plants, they take light energy and convert that into chemical energy, using this energy from the sun to build organic molecules. We'll see this in a later chapter. But the thing to keep in mind is that all we're doing is shuffling stuff around. In any chemical reaction, in any big pathway, all throughout this quarter, always keep this in the back of your head, that we are never magically creating energy out of nowhere. It never appears out of nowhere. It always has to come from somewhere, and it always has to go somewhere as well. All we're doing is converting stuff from one form to another, because energy cannot be created or destroyed. Okay. Again, that's all there is for thermodynamics in this intro biology class. Now that we have that, we can start talking about chemical reactions. So we can break chemical reactions down into two basic types. And I think it's best to look at these in graph form. So if we just look at the basics of this graph, we're looking at this says Gibbs free energy. It just means how much total energy is in these components. And the axis down here is time. So this is a type of chemical reaction where our starting materials are at a certain energy level, and our products, when we finish this chemical reaction, are at a lower energy level. In other words, this is a destruction reaction. This is a breakdown reaction. We're taking something complicated with a lot of energy, and we're turning it into something that is less complicated with fewer bonds and just has less, smaller amount of total energy. Of course, thermodynamics, energy can't be created or destroyed, so we're going from high energy to low energy components. There is energy that is released from this reaction. So if we want to put this in words, this is called an exergonic reaction, where products are at lower energy than the reactants, the starting materials. And this type of reaction releases energy into the surroundings. You could imagine the other of these two types of chemical reactions is the exact opposite. This is called an endergonic reaction, and the exact opposite. You start with low energy things, you end up with high energy things. Energy cannot be created or destroyed, so if you want to go from low energy to high energy, you need to put energy into this reaction. So endergonic reactions are where products are at higher energy than the reactants, and this type of reaction requires an input of energy. Now, real quick side note, not for the first time, not for the last time. We got two different things that endergonic and exergonic that are very similar sounding and are very easy to mix up on a multiple choice exam. My dumb way of remembering which one is which is exergonic reactions release energy. If you crack open a thesaurus and search for release or let out and try to find some other word, some synonym for release, one word for that is extrude or exude. Exude just means the same thing as letting forth, releasing. So I don't know if you've got a good vocabulary or like that, you could think of exergonic reactions exuding energy, releasing energy. And then this is the other one. I can't think of a synonym for requiring energy input that starts with end, but exergonics exude energy, endergonics are the other one. So not writing any of that down, just one way to remember the difference, because these will be next to one another on multiple choice questions. Know which one is which. Okay. Now, the exergonic reactions here release energy, and the endergonic reactions require energy to actually happen. Unsurprisingly, you could put the two of these together. You can couple these reactions to happen together at the same time, so these two can be coupled such that the energy released from this reaction serves as the energy input required for this reaction. We're going to see this all the time in cells and biological systems, coupling these two types of reactions to one another. Now, there's one more thing about these graphs that I've kind of ignored up until now, and that is this bump right here. So yeah, we're starting with high energy and going to low energy in exergonic and going from low to high in endergonic, but there's this extra hump present in here. Even though overall we're going from high to low, first we have to go even higher. And even though we're going from low to high, we have to get past this. We have to go to an even higher energy level before we can get to our end products. This energy hump right here, which we see in both of these graphs, is called activation energy. So we can define this here. Both of these chemical reactions, both endergonic and exergonic, need to overcome an activation energy, which you could define as simply an additional energy input in order to proceed. And again, I said even exergonic because exergonics, they're releasing energy. You're sending energy out. Even though by the end of this you are going to be releasing energy, you do have to put in some amount of energy first before you can release this. Now, this energy hump is very important because it prevents molecules from just spontaneously breaking down. If there was no energy hump here, every single complicated molecule ever could just slide down to low energy products and release energy. This doesn't happen because, there's this hump getting in the way. So yeah, let's take a look at this stack of wood here. Wood has lots of potential chemical energy in it, lots of high energy bonds holding together all of the chemical compounds in this wood. The reason why this wood is just sitting there and not spontaneously falling apart into simpler molecules and releasing a bunch of energy is because it can't get over this activation energy. These molecules are just sitting there and they're not getting over that activation energy. They're staying put where they are and not proceeding to low energy products. So activation energy, this prevents reactions from occurring spontaneously. Now, that's great for keeping things stable, but what if we want this reaction to go forward? What if we want these high energy reactants to slide down to lower energy products and release a bunch of energy? What if we want to destroy this wood and get energy out of it? Well, one way that we can get past this hump and cause this reaction to actually go forward is to add some energy to it. And yes, this is the type of reaction that's going to release energy, but I guess there's a saying in the business world, you got to spend money to make money, and that's kind of what can go on here. If you spend energy here to get past this hump, you can let the reaction proceed and release energy. You could probably see how this works since this already kind of looks like a campfire here. If you add heat energy to a chemical reaction, that can be used to overcome the activation energy and cause that reaction to go forward. So add some heat to this, inert pile of wood. It's going to overcome the activation energy. And that's what a fire is. It's breaking these complicated molecules down into carbon dioxide and water, and it's releasing a whole bunch of heat as these reactions go from high energy to low energy. Now, this works in a campfire, or an oven or whatever, but this adding of heat is not something that's feasible inside of living things. You can't do this in a cell. So I want to point out that this is a way to make this happen, but it's not what we do in living things. So what do we do as living things to make these reactions actually happen? Well, we use chemical compounds called catalysts. So here's our familiar looking graph for an exergonic reaction, high energy reactants, lower energy products. The purple line here is our normal reaction we've been seeing. When a catalyst is used, that's what the green line is. So a catalyst is something that will, as it says in the title, decrease the activation energy, lower the activation energy. Importantly, a catalyst will not remove the activation energy. The hump is always going to be there. Catalysts simply lower it. Catalysts decrease activation energy. And we're seeing this here in an exergonic reaction. Catalysts can work on endergonic reactions as well. Again, uncatalyzed here in purple, catalyzed reaction here in the dashed red lines. The activation energy is still there, it's just not as much. So how do they do this? How do you magically just make this graph look differently? Well, what a catalyst does is a catalyst holds the reactant or reactants, the starting materials here, and strains their chemical bonds. That is what is going to cause the activation energy to lower. I'm going to skip ahead a few slides to something to show what this can look like. Here is our catalyst. Here is our starting material, and it is held by the catalyst and hugged and strained without having to spend any energy. The catalyst is not expending any energy to do this. It's just changing its shape a little bit to strain the bonds a little bit in this starting material, effectively lowering that activation energy. So catalysts hold reactants, strain their chemical bonds. Catalysts lower activation energy. And like we saw with these two graphs, these can be used to facilitate exergonic reactions or endergonic reactions. Now, catalyst is a very broad term. There are a lot of things that catalyze chemical reactions. We're going to get more specific now and talk about the kind of catalysts that are biological in nature. There's a special name for these things. Biological catalysts, and side note, almost all these things are proteins. Protein catalysts are called enzymes. So an enzyme is a type of catalyst that is biological in nature. It's almost always a protein. So again, enzymes are a type of catalyst. They do all the stuff we just talked about with activation energy, but they are usually proteins. So let's talk about the process that goes on when a catalyst or an enzyme does its thing. So here's our very cartoony kind of overview of enzyme function, enzyme catalysis. And it begins with our enzyme, it's the gray blob here. It begins with our enzyme binding to the starting materials. So we've got some special terminology here. Our starting materials in an enzyme catalyzed reaction are called the substrate or substrates. Now, this particular example is showing one chemical compound being broken into two. So sometimes there's just a single substrate binding to the enzyme. But remember, there are also enzymes that catalyze reactions like this, building reactions, endergonic reactions. Sometimes, even though it's not pictured in the example here, there are enzymes that bind a couple of substrates and will connect them together. So that's why I'm going to hit you with the parenthetical S here. Our step number one of enzyme catalysis, the substrate or substrates, depending on what's going on here, bind at the enzyme's active site. That's another piece of terminology here. It's a special little pocket that is built for the substrate or substrates to bind. Again, we know there's going to be the hug here. We know it's going to strain the chemical bonds. It makes sense that there would have to be a special little pocket. So terminology here, substrates bind at active site. In doing so, they form what is called the enzyme substrate complex. And this brings us to, this figure that I showed just a minute ago. Substrate, active site, bind. Now we have enzyme substrate complex. So this is a very specific binding event. So this pocket right here is built just for this substrate. If there's another chemical compound out there that's kind of similar to this, it probably won't fit in this active site. This is sometimes compared to the specificity between a lock and a key. You may have a similar shaped key to this lock, but if it ain't exactly the same, it's not going to fit. You need a different lock for that key, different key for that lock. Enzymes are very specific for what substrate or substrates they bind. So the cell's going to have a lot of different enzymes in it because there are a lot of different substrates out there. But yeah, the specificity is very important to note here. Now, the part where this lock and key analogy breaks down a little bit is that once this substrate binds to the active site, the enzyme itself actually kind of changes its shape just a little bit. This hug that you could see here, the term that I use is hug, is called induced fit. So once this binding has occurred, the enzyme changes its shape slightly to better fit that substrate or substrates. Again, this hug, the formal term for this is induced fit. This is where we have straining of the chemical bonds in the substrate or substrates. This induced fit, this hug, is what lowers the activation energy of this reaction. Now, as I noted, when I showed this figure earlier, this does not remove the activation energy. It's still there. However, if this activation energy is lowered sufficiently, the ambient heat energy, so there's energy all around us all the time. If this is lowered enough, in an exergonic reaction, the ambient heat energy can be enough to overcome that activation energy. So let me give you an example of this. So we looked at a pile of wood earlier and saw how we could add heat energy to this to make these chemical reactions go forward. Let's look at that pile of wood again. So there are certain animals out there that make a living eating piles of wood like this. Here is a protist, trichonympha, I believe, that lives inside of the gut of termites. Termites are going to chomp on this wood. The microbes inside their gut are going to break down this wood, not by lighting it on fire, but through enzymes. They have an enzyme that is going to bind the cellulose in this wood, strain the chemical bonds in this, and cause it to break apart. There's enough heat inside the termite, inside the world around the termite, that once this activation energy is lowered enough, the ambient heat energy is going to make this go forward and is going to break down this chemical compound so they can get energy out of it. So yeah, this is just showing how ambient heat energy can overcome this activation energy if you lower it enough. For an exergonic reaction, an endergonic reaction, you still need to put energy into it to create higher energy products. The ambient heat energy is not enough to make this go forward, but the energy that you put in is not as much as you would normally have to. It's just less energy you have to put in to this type of reaction. Okay, so this was number 2, the hug, the induced fit, the straining. Step number 3, the chemical reaction occurs. Again, whatever it is, it actually happens. So let me go back to this cartoon here. So substrate, active site, enzyme substrate complex. There's the induced fit. You can see how the enzyme has a slightly different shape here than it did before. And then, here's the chemical reaction. Now there's one bond in this example that is broken apart. This is different than what it was before. And next, the products are released. So the product, again, that parenthetical S, because sometimes it's one product, sometimes it's 2 products. The product or products are released from the enzyme. Now, this might seem like the last step here in enzyme function, but there's a very important final step. The enzyme goes back to how it was before. Understand that throughout this process, the enzyme does not permanently change. The substrate is changed into products, but the enzyme, it does induced fit, it changes its shape slightly, but by the time it's finished catalyzing this reaction, it's the same shape it was in when it started. This means enzymes are going to do their chemical reaction over and over and over and over and over again. As long as there's substrate around in their environment, enzymes are going to continuously catalyze this reaction and churn out products. So our very important 5th step here is that the enzyme assumes its original shape. It is not permanently altered by this reaction. It is very much free to work again. So enzymes are not one-time use only molecules. They're going to do their thing over and over and over and over again. Okay, so this is, these 5 steps, and this very good overview, single slide overview, is a good description of how enzymes work normally. However, there are a few things that can mess with this normal functionality of an enzyme. One of these things is something called an enzyme inhibitor. As the name implies, enzyme inhibitors inhibit enzymes. They reduce an enzyme's function. They make the enzyme work not as well as it is supposed to, or not at all. Some of these inhibitors work by gumming up the active site, preventing the substrate from coming in. This is a dead enzyme. It can't do anything. The inhibitor is in the way. Other inhibitors don't bind to the active site. They bind somewhere else, but they change the shape of the overall enzyme so that substrate cannot bind, and this is an ineffective enzyme. So a lot of details labeled here, but the take home is enzyme inhibitors are chemical compounds that reduce an enzyme's function. A lot of toxins and poisons, we'll mention a couple of these in the next chapter, I think. Things like arsenic and cyanide, things that are very deadly to us, are inhibitors that shut down very, very, very important enzymes. So, inhibitors shut down enzymes. There's another one that does the exact opposite. Chemical compounds called enzyme activators increase an enzyme's function. So enzyme activators bind to an enzyme, change its shape a little bit, and give it a better active site, an active site that is more effective at binding substrate. This is going to make these more effective enzymes. I don't know if there are any toxins or poisons that act as activators. I'm sure there are some out there. None come to mind right now, but there are plenty of activators that the cell itself makes as a way to, tune up the level of certain enzymes and then turn down the level of enzymes. So your own cells are making inhibitors and activators to just control the level of an enzyme's functionality. But all I'm writing for this, activators increase things. Now, there are 2 other components. So we got inhibitors that can affect enzymes, activators that can affect enzymes, and then things called cofactors and coenzymes. So these things are optional. That's why I say some enzymes. Some enzymes require cofactors. Some enzymes require coenzymes. Some enzymes require both of these things, and some enzymes don't need either of these things. So that's what I mean by cofactors and or coenzymes. These are optional. Some need one, some need the other, some need both. So what are these things, and what are their definitions? They're both in the key terms, obviously. So cofactors are small ions. Don't memorize this table, but cofactors are things like zinc and iron and magnesium. These are things that, we know that we need a little bit in our diet. If you take vitamins and supplements, there's going to be some zinc and iron in those things. The reason why we need these charged atoms is because we've got enzymes that require them. So I believe the key terms defines a cofactor as an inorganic ion that is required for optimal enzyme activity. Again, the phrase inorganic ion is very important here for cofactor. These are not big, complicated molecules. These are single atoms, and they have a charge to them. Inorganic ions. In contrast, coenzymes are big, complicated organic molecules. These are, again, don't memorize these, but these are examples of coenzymes. Many of these are vitamins: vitamin A, vitamin B, vitamin E, vitamin D, vitamin C. These are, once again, things that you need as part of your diet because you got plenty of enzymes that require these specific molecules in order to function properly. The key terms definition of a coenzyme is a small organic molecule that is required for optimal enzyme activity. So the coenzymes are organic molecules, lots of carbon, 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 carbon here. The cofactors, these are completely inorganic. They're just a single charged atom. So that's the difference between cofactor and coenzyme. And again, some enzymes need them. Okay. Now, each enzyme only performs a single chemical reaction. If I can go back to this slide, this enzyme is breaking this one single chemical bond in this substrate to create two products. If you want to build something very complicated, let's look at glucose here. Look at all these chemical bonds in glucose. That's quite a lot of bonds holding this together. If you want to build this from scratch, you cannot do that with a single enzyme. If you want to build something complicated, you're going to need a bunch of enzymes working together in an assembly line, with each enzyme performing one function. Yeah, here's an auto factory, some workers here building engines, and yeah, each worker is gonna, I guess they'd be robots in modern times, but in this old-timey black and white photo, each worker is performing one modification to this as it moves on to the next worker. That's what we got to do in biology if we want to build something complicated. Or if we want to break down something complicated, if we want a disassembly line, if we want to disassemble this complicated molecule into individual parts, we're also going to need a line of enzymes to do this. It takes a series of steps to build or break down a complex molecule. This series of steps, with each step being carried out by a single enzyme, is called a metabolic pathway. So there are assembly line metabolic pathways and there are disassembly line metabolic pathways. Yep, those are going to be our two types. Let's start with the building one. So two types of metabolic pathways. One of these is called an anabolic pathway. Anabolic pathways build complex molecules. These are assembly line pathways. And yeah, taking smaller things in a series of steps, connecting them together and building something that's more complicated. Anabolic pathway. And importantly, this requires energy. We are creating a lot of chemical bonds as we make this bigger, more complicated thing. Anabolic pathways require an input of energy. Okay, the other one is, as you could imagine, the exact opposite. The other type of metabolic pathway is called a catabolic pathway. Catabolic pathways break down complex molecules and release energy. So, you got a big complicated molecule, you're going to break it into individual things. It's going to take a lot of individual steps to do that, and you're going to release a bunch of energy as a result. Anabolics build, catabolics break down. All together, if we look at all the anabolic and catabolic reactions going on in the cell, we refer to this as metabolism, just talking about all of the chemical pathways, all the metabolic pathways going on are collectively referred to as metabolism. Which, I almost forgot, is defined in the key terms. All the chemical reactions that take place inside cells, including anabolic and catabolic pathways. So metabolism, just referring to all these things together. Now, a lot of students get mixed up on how anabolic and catabolic are different from endergonic and exergonic. Because, for here, I'm saying anabolic requires energy input, catabolic releases energy. I said the same two things when I was describing these reactions. The difference between anabolic and catabolic and endergonic and exergonic is that these terms describe a single chemical reaction. A single chemical reaction is either exergonic or endergonic, whether it releases energy or requires an energy input. The terms anabolic and catabolic refer to big pathways. So yes, an anabolic pathway is going to have a lot of endergonic reactions as part of that pathway. A catabolic pathway is going to have a lot of exergonic reactions going on in it. These are just bigger picture terms that refer to those large pathways. These terms refer to individual chemical reactions. So yeah, just to clear up this common confusion, these are not redundant terms. These refer to pathways. These refer to individual chemical reactions. All right, let's close out this chapter by talking not about a big, complicated enzyme or pathway, but by talking about a single molecule that is incredibly important when we talk about energy in the cell. We're going to talk about something called adenosine triphosphate. That's quite a mouthful. It's usually referred to simply as ATP, adenosine triphosphate. Now, let's take a look at this. And like I keep saying, you don't have to memorize this exact structure, but there are several things to point out here that are important. First of all, this should look familiar to you from an earlier chapter. We've got a ribose sugar down here. We have this two-ring structure here with a lot of nitrogen, a nitrogenous base, if you will, and it looks like a phosphate here as well. Yeah, this is a nucleotide. This is a building block of DNA or RNA, or since it's a ribose here and not a deoxyribose, technically it's a building block of RNA. But yeah, ATP is a nucleotide. One of its jobs is a building block constructing RNA. But that's not what we're here to talk about. Here we're here to talk about ATP's other very important job as an energy storage molecule. ATP has a very convenient couple of chemical bonds. So, as I said near the beginning of this chapter, chemical bonds store energy. Breaking chemical bonds releases energy. So technically, any chemical bond is a source of potential chemical energy. But, as it turns out, this bond right here, connecting this phosphate to this phosphate. Oh yeah, by the way, it's not just one phosphate, it's got 3 phosphate groups, hence the name triphosphate. The chemical bond holding together this phosphate and this phosphate is a really high energy bond. Breaking this bond releases a lot of energy. And there's another one right here, connecting this phosphate to this phosphate. Importantly, the bond between this final phosphate and the sugar is not a bond we care about. It's not one of those high energy bonds. So there are 3 phosphates here in this chemical compound, but there are only 2 high energy bonds. The ones between these 2 phosphates and the one between these 2 phosphates. So ATP has chemical potential energy 2. I can't remember my test that I wrote, but I may try to trick you by asking you how many chemical bonds this has that are important. There are 3 phosphate groups here, obviously, triphosphates right there in the name, but there are only 2 high energy bonds present within this molecule. They're the bonds between these 3 phosphate groups. Again, these are very convenient bonds. Breaking them releases energy. That is an exergonic reaction. Because this is such a relatively small, convenient, easy to work with molecule, this reaction, this exergonic reaction that occurs when you break off one of those phosphate groups is very easily coupled to other endergonic reactions to provide the energy they need. So in the cell, we're going to see this in the next several chapters. If you've got an endergonic reaction that needs to happen, if you're building something, if you're trying to create something more complicated, you're trying to make chemical bonds and you need an input of energy, you just break down ATP next to it. As part of the same enzyme, you just grab an ATP, break off one of those phosphate groups. That's going to be an exergonic reaction. That's going to release energy. That's going to give you the energy you need. So if we want to kind of draw this out here, we could call ATP the energy currency of the cell. Again, almost any time the cell needs energy, they're going to get it from ATP. So here's ATP. You break off one of those phosphates, the P subscript lowercase I, the PI just refers to a loose phosphate. So when you've ripped off one of these phosphates here, you've got an inorganic phosphate, a loose phosphate, and you no longer have adenosine triphosphate, you have adenosine diphosphate, only 2 phosphates left. But this reaction releases a bunch of good energy. Remember, there are 2 high energy bonds, so we can do this a second time. If you take ADP and you rip off this phosphate from the first phosphate, you're going to have another loose phosphate, and you're going to have a molecule called adenosine monophosphate. Only 1 phosphate left, and that, of course, also releases a bunch of energy. And that's it, just these 2. This third phosphate, its connection with the sugar is not a high energy bond here. I mean, it can be broken down, but it's not as convenient. It's not really used as energy currency. There are 3 phosphates, but there are only 2 high energy bonds that can be exploited for energy here. So there is a problem with ATP. Again, as convenient as this is, as great of a job as it does of being energy currency and providing energy for endergonic reactions, the problem is that it doesn't have a good shelf life. ATP is not particularly stable, so we use this for everything in the cell, but it breaks down on its own. Like I said, unstable, bad shelf life. So what we need to do as an organism, as a cell, as a living thing, is we need to store our energy long-term in other molecules. Things we've already brought up in previous chapters. Things like fats, triglycerides, things like carbohydrates, monosaccharides, disaccharides, polysaccharides. Those things have a great shelf life. We're going to store our energy long-term in these other molecules, and then when we actually need energy, when we actually need the power to make endergonic reactions happen, we need to convert the energy in these long-term storage molecules into ATP so that we can actually use it. This process of shuffling around energy, of converting it from long-term storage to usable ATP, is going to be the topic of our next chapter, which I will begin in the next recorded lecture. So yeah, a lot more energy to come, but this chapter on metabolism really laid down some groundwork, some important terminology. We'll talk about very specific metabolic pathways in the next couple of chapters. But yeah, this is the end of chapter 6. This is the end of recorded lecture 2.2.